Hello, I'm Dr. Christopher Liebman, the Commercial Director and Co-founder of Chromacity, a manufacturer of ultra-short pulse lasers and tunable OPO systems. Today, I'm going to be talking about the adoption of ultrafast lasers for multiple biophotonics applications, looking at key factors that are driving development. Before we begin, I'd like to thank the organisers very much for inviting me to speak. I'm just sorry that we can't meet at Photonics West this year. The role of photonics in healthcare is always pushing the boundaries of life sciences imaging, and now increasingly more economical light sources are required that deliver the necessary technical performance characteristics. In this presentation, I will talk about the evolution of ultrafast sources over the last few years and demonstrate how important factors such as integration, reliability and robustness are fostering new opportunities for ultra-short pulse lasers to be adopted for a wide range of life sciences imaging applications. And I'll be giving some examples along the way. So historically, the multi-photon imaging community relied heavily on titanium sapphire lasers. These were diode-pumped solid-state systems and typically required a laser physicist to operate these systems, and you can see this typical kind of setup here on the left. They were bulky, temperamental, and took hours to warm up and expensive to run and purchase. In saying that, they did generate very short sub-100 femtosecond pulses and correspondingly broad bandwidths, making them ideal to excite a wide range of fluorescent markers. Over the years, significant improvements have been made to electronically control the cavity to make these systems significantly more user-friendly. Although, as you see on the right here, they do still have a relatively large footprint and still come at a relatively high price. For research that requires excitation of multiple markers, there is still very much a place for these kind of broad bandwidth light sources. So why has the life sciences imaging community looked for an alternative technology? Well, fiber-based laser architectures are inherently more efficient, stable and robust. This efficiency leads to smaller thermal demands, therefore many of today's laser-based systems don't require water cooling at all. Since the intracavity beam is confined within a fibre, the stability of these systems is significantly better than free space laser cavities. This leads to less breakdowns and maintenance issues. Therefore, there's a corresponding reduction in ongoing warranty costs. The simplicity of operation means that imaging can be carried out more rapidly, therefore leading to faster research outputs. All of these aspects lead to a lower cost of ownership throughout the lifetime of the laser, and more time to dedicate towards the image acquisition without the need of a laser physicist. But there's more to it than that, because there are a whole range of imaging modalities that demand very specific technical requirements. And we at Chromacity have demonstrated a number of these kinds of capabilities, either through two-photon imaging of liver samples, as you see here on the left, or multimodal imaging through two-photon and SHG imaging of, I think it was collagen fibres sitting over the top of kidneys, as you see here in the centre. And then on the right-hand side, we've also demonstrated two-photon light sheet capabilities where we can image zebrafish embryos. So all of these different kinds of modalities have slightly different requirements. A lot of the work that can be achieved depends very much on the fluorescent probes that need to be excited. One of the strengths of sapphire lasers is that they can span the excitation bandwidths of many of these probes. But much of today's research doesn't require the excitation of multiple markers. In fact, there's generally two main wavelength regions that are targeted. For two-photon and SHG imaging in particular, this band is around about the 1000 to 1100 nanometer region, and also the sort of 920 to 980 nanometer region. Fiber-based technologies started their development at around about 1040 to 1060 nanometers. But there is now a new generation of sources coming onto the market that covers that secondary 920 to 950 nanometer region. The beauty of these fluorescent probes is that they have very large excitation bandwidths as well, as can be seen by the graph here on the left. This allows a range of labels to be excited by a fixed wavelength source. Indeed, we worked with the Institute of Genetics and Molecular Medicine here in Edinburgh to demonstrate this capability using our Chromacity 1040 femtosecond laser source. This image here on the right used our laser to image liver cells that were labelled with RFP, YFP and GFP simultaneously. And here you can see that the three separate channels are overlaid with each other. Only about 150 milliwatts of laser light was actually incident on the Galvo scanning mirrors within this Nikon microscope. And because of this two-photon technique, we could image at depth. But if we now look at these three individual channels separated out as illustrated on the left here, you can see that the uptake of GFP within the nuclei of the cells was actually very poor. This was due to the particular cell line that we were imaging. But it also doesn't help that the wavelength of our source is very much at the edge of excitation for GFP. In saying that, several green fluorescent proteins have a high fluorescence quantum yield, which allows us to demonstrate this important capability of exciting multiple markers from a fixed wavelength source. Other capabilities include the ability to demonstrate electrophysiology measurements, particularly in cardiac and neural tissue. This frequently involves recording membrane potentials and calcium transients via multiphoton processes, as indicated by the image on the right here. For this purpose, a broad range of voltage and calcium-sensitive fluorophores have been specifically developed. However, these processes are typically in the second infrared imaging window, typically across the 1 to 1.3 micron region, where the power of sapphire systems typically falls away dramatically, but where multiphoton microscopy has been demonstrated as a versatile tool in the life sciences for many structural and functional applications both in situ and in vivo, including electrophysiology, calcium imaging, blood flow and immune cell trafficking in the heart in vivo. We worked with cardiologists at the University of Glasgow to demonstrate this capability of our system in this important imaging field. We were keen to understand how our Chromacity 1040 fixed wavelength source compared against a standard sapphire system for enhanced fluorescence-based functional measurements at depth in the intact heart. On the left is a schematic of the setup, but more importantly on the right you can see how the experiment is carried out within a very confined space. The sapphire systems take up much of the space on the top left hand of the lab bench. The power supply and water chiller takes up much of the space underneath the bench. In comparison, our system, circled in orange, has a much more compact footprint, as does its power supply. The significantly small footprint is now commonplace for many fibre laser manufacturers. Integrating into the Zeiss LSM microscope system was simple, and we yielded results quickly as a consequence of this. When comparing our systems to the likes of a TISAF, it was clear that we could meet the technical demands of deep tissue optical electrophysiological imaging. In addition, we had the ability to fine-tune the dispersion characteristics of our system to account for the dispersion through the microscope objective lenses, thus maximising fluorescence at the sample plane. In this short demonstration, we were able to record action potentials at depths of over 250 microns, whereas the Sapphire was only able to reach 200 microns. This is a consequence of the TISAF powers tailing off at and around 1 micron. Even with the losses that are incurred through a standard microscope system, the Chromacity laser has the ability to deliver more than 400 milliwatts at the sample plane, way in excess of the 75 milliwatts used to obtain these images as illustrated here. While 400 milliwatts is in excess of what biological tissues can tolerate, we believe that increasing the power beyond 75 milliwatts at deeper transmural tissue planes would increase the range of imaging of the system even further than 250 microns that we demonstrated, whilst still tolerating energy limits for the intact cardiac tissue. This is one of the benefits of being able to generate high average powers from a fixed wavelength femtosecond fibre laser system. To give you a sense of our capabilities, I include this slide which details our typical laser parameters. So what you're looking at here is an image of the laser system itself, along with an autocorrelation trace demonstrating the ultra-short pulse nature of the light source. And then further to the right is the very good beam quality that we get from our fibre laser system, making it very easy to integrate into a microscope setup. And on the right-hand side there is the very simple web-based user interface that can be used to operate all of our laser systems. We offer typically 3 different power versions of our 1 micron source. So we starting at half a watt, 500 milliwatts of laser light, and then moving all the way up to greater than 3.5 watts, which is often ideal for those researchers looking at using SLMs and holographic devices that are high lossy, therefore you need the high average power. But it's not just the laser performance characteristics that are important. As I indicated in the earlier slide, we had to optimize the dispersion through the optical system by pre-chirping the pulse. This type of dispersion management is becoming commonplace within the laser head of fibre-based laser systems. Chromacity can tune the pulse duration typically anywhere between 1 picosecond and sub 150 femtoseconds. The other main feature that is becoming more important is the ability to vary the power of the laser system on microsecond timescales. This is to help to avoid bleaching cells as the laser beam is raster scanned across samples. More and more laser manufacturers are now offering this as a built-in capability. There's also the need for higher average power systems, particularly when implementing beam shaping, spatial light modulators or holographic techniques. The losses incurred in these imaging modalities require high average laser systems in order to deliver enough light to the sample plane. Hence why Chromacity developed its high power capability, where we can deliver more than 3.5 watts. It's clear that there are a whole host of life science imaging applications that can make use of these fixed wavelength sources. One of the exciting applications we've been involved in is looking at the heating of hybrid gold iron oxide nanoparticles in biological media en route to pushing towards developing targeted therapies against pancreatic cancer. Here, hybrid gold iron oxide nanoparticles have been shown to hold great potential for heat-triggered drug delivery. The magnetic core of these particles enables magnetic resonance imaging, whilst the gold surface coating can act as a localized heat source after laser irradiation by exploitation of the surface plasma resonance. We carried out a comparative study looking at the differences between our fixed wavelength femtosecond 1 micron source against the standard nanosecond 1064 system that the academic was using beforehand. And in this work, human pancreatic cancer cells were cultured within 5-week-old female mice. 2 different hybrid nanoparticle concentrations of either 50 micrograms per milliliter or 5 micrograms per milliliter were irradiated within the tumours. Thermal rise and heat dissipation was measured in xenograft-bearing cadavers, and the hybrid nanoparticles were injected intratumorally. What we were able to ascertain from these measurements is that with the larger concentration of 50 micrograms per milliliter, we could see temperature differences of up to 80 degrees centigrade, and at the lower concentrations, temperature increases of 26 degrees. Figure B here shows the heat dissipation in the tumour tissue, and here we observe that at 5 millimeter radiuses from the irradiation point, the majority of heat has been lost, with only small thermal changes beyond this point. With the previous laser system that the customer was using, no stable heating was observed when irradiating the nanoparticles at the lower concentration of 5 micrograms per milliliter. However, in this study, in the xenografts in particular, those injected with the lower concentration experienced 27 degrees temperature increases when pumped by our Chromacity 1040 system. Additionally, there was an 8-fold increase in temperature when using the higher concentration compared with the previous 1 micron source that the customer was using. These important findings show that for biomedical applications, more than a 10-fold reduction in nanoparticle concentrations could be administered in order to experience similar heating effects. This would obviously reduce any question over the toxic burden of nanocarrier or long-term accumulation within the tissue. This capability would enable either localized tumor ablation or thermal triggering drug delivery, which is highly localized to the laser irradiation point, thus minimizing impact on the surrounding tissue. Clearly, this is ongoing research, but something that we're very excited to be involved in. And finally, I'd like to mention the importance of being able to easily integrate these laser sources into a range of microscope setups. In order to do this, we need to consider important laser parameters like beam shape, m² values and divergence. They all play a part in ensuring ease of setup and ultimately ease of use. With home-built setups, it's particularly important to have discussions with the laser manufacturer to ensure that the correct technical specifications are suitable for your particular imaging modality. In conclusion, fibre-based systems are gaining more and more traction in the life sciences sector, and as a consequence of their technical capabilities, additional functionality and low cost of ownership. Along with the advent of new laser lines like 920 nanometers to complement the more traditional 1 micron sources, it's clear that these systems are going to increase the ability to create exciting discoveries from an increasing number of labs. This is something Chromasti is really excited to be part of. So many thanks for watching, and I look forward to following up with you with any queries you may have. And I hope to meet with you in person in San Francisco next year. Many thanks and all the best.